Chapter 41. Small Intestine, Functional Anatomy. Small intestine is the part of gastrointestinal, GI, tract, extending between the pyloric sphincter of stomach and ileocecal valve, which opens into large intestine. It is called small intestine because of its small diameter, compared to that of the large intestine. But it is longer than large intestine. Its length is about 6 meters. Important function of small intestine is absorption. Maximum absorption of digested food products takes place in small intestine. Small intestine consists of 3 portions: proximal part known as duodenum, middle part known as jejunum, distal part known as ileum. Wall of the small intestine has all the 4 layers as in stomach, chapter 36. Intestinal villi and glands of small intestine. Intestinal villi. Mucous membrane of small intestine is covered by minute projections called villi. The height of villi is about 1 mm and the diameter is less than 1 mm. Villi are lined by columnar cells, which are called enterocytes. Each enterocyte gives rise to hair-like projections called microvilli. Villi and microvilli increase the surface area of mucous membrane by many folds. Within each villus, there is a central channel called lacteal, which opens into lymphatic vessels. It contains blood vessels also. Crypts of Lieberkühn or intestinal glands. Crypts of Lieberkühn or intestinal glands are simple tubular glands of intestine. Intestinal glands do not penetrate the muscularis mucosa of the intestinal wall, but open into the lumen of intestine between the villi. Intestinal glands are lined by columnar cells. Lining of each gland is continuous with epithelial lining of the villi, Fig 41.1. Epithelial cells lining the intestinal glands undergo division by mitosis at a faster rate. Newly formed cells push the older cells upward over the lining of villi. These cells which move to villi are called enterocytes. Enterocytes secrete the enzymes. Old enterocytes are continuously shed into lumen along with enzymes. Types of cells interposed between columnar cells of intestinal glands: argentophane cells or enterochromophane cells, which secrete intrinsic factor of Castle. Goblet cells, which secrete mucus. Paneth cells, which secrete the cytokines called defensins. Brunner glands. In addition to intestinal glands, the first part of duodenum contains some mucus glands, which are called Brunner glands. These glands penetrate muscularis mucosa and extend up to the submucous coat of the intestinal wall. Brunner glands open into the lumen of intestine directly. Brunner glands secretes mucus and traces of enzymes. Properties and composition of Succus Antericus. Secretion from small intestine is called Succus Antericus. Properties of Succus Antericus. Volume, 1800 ml/day. Reaction, alkaline. pH 8.3. Composition of Succus Antericus. Succus Antericus contains water, 99.5%, and solids, 0.5%. Solids include organic and inorganic substances, Fig 41.2. Bicarbonate concentration is slightly high in Succus Antericus. Functions of Succus Antericus. 1. Digestive function. Enzymes of Succus Antericus act on the partially digested food and convert them into final digestive products. Enzymes are produced and released into Succus Antericus by enterocytes of the villi. Proteolytic enzymes. Proteolytic enzymes present in Succus Antericus are the peptidases, which are given in Fig 41.2. These peptidases convert peptides into amino acids. Amylolytic enzymes. Amylolytic enzymes of Succus Antericus are listed in Fig 41.2. Lactase, sucrase, and maltase convert the disaccharides, lactose, sucrose, and maltose, into 2 molecules of monosaccharides, Table 41.1. Dextrinase converts dextrin, maltose, and maltriose into glucose. Trihalase or trihalose glucohydrolase causes hydrolysis of trihalose, carbohydrate present in mushrooms and yeast, and converts it into glucose. Lipolytic enzyme. Intestinal lipase acts on triglycerides and converts them into fatty acids. 2. Protective function. Mucus present in the succus entericus protects the intestinal wall from the acid chyme, which enters the intestine from stomach, thereby it prevents the intestinal ulcer. Defensins secreted by Paneth cells of intestinal glands are the antimicrobial peptides. These peptides are called natural peptide antibiotics because of their role in killing the phagocytosed bacteria. 3. Activator function. Enterokinase present in intestinal juice activates trypsinogen into trypsin. Trypsin, in turn activates other enzymes, chapter 39. 4. Hemopoietic function. Intrinsic factor of Castle present in the intestine plays an important role in erythropoiesis, chapter 10. It is necessary for the absorption of vitamin B12. 5. Hydrolytic process. Intestinal juice helps in all the enzymatic reactions of digestion. Functions of small intestine. 1. Mechanical function. Mixing movements of small intestine help in the thorough mixing of chyme with the digestive juices like Succus entericus, pancreatic juice, and bile. 2. Secretory function. Small intestine secretes Succus entericus, enterokinase, and the GI hormones. 3. Hormonal function. Small intestine secretes many GI hormones such as secretin, cholecystokinin, etc. These hormones regulate the movement of GI tract and secretory activities of small intestine and pancreas, chapter 44. 4. Digestive function. Refer functions of Succus entericus. 5. Activator function. Refer functions of Succus entericus. 6. Hemopoietic function. Refer functions of Succus entericus. 7. Hydrolytic function. Refer functions of Succus entericus. 8. Absorptive functions. Presence of villi and microvilli in small intestinal mucosa increases the surface area of mucosa. This facilitates the absorptive function of intestine. Digested products of foodstuffs, proteins, carbohydrates, fats, and other nutritive substances such as vitamins, minerals and water are absorbed mostly in small intestine. From the lumen of intestine, these substances pass through lacteal of villi, cross the mucosa and enter the blood directly or through lymphatics. Absorption of carbohydrates. Absorption of proteins. Absorption of fats. Absorption of water and minerals. In small intestine, sodium is absorbed actively. It is responsible for absorption of glucose, amino acids, and other substances by means of sodium cotransport. Water moves in or out of the intestinal lumen until the osmotic pressure of intestinal contents becomes equal to that of plasma. In ileum, chloride ion is actively absorbed in exchange for bicarbonate. The significance of this exchange is not known. Calcium is actively absorbed mostly in upper part of small intestine. Absorption of vitamins. Most of the vitamins are absorbed in upper part of small intestine and vitamin B12 is absorbed in ileum. Absorption of water-soluble vitamins is faster than fat-soluble vitamins. Regulation of secretion of Succus entericus. Secretion of Succus entericus is regulated by both nervous and hormonal mechanisms. Nervous regulation. Stimulation of parasympathetic nerves causes vasodilatation and increases the secretion of Succus entericus. Stimulation of sympathetic nerves causes vasoconstriction and decreases the secretion of Succus entericus. But, the role of these nerves in the regulation of intestinal secretion in physiological conditions is uncertain. However, the local nervous reflexes play an important role in increasing the secretion of intestinal juice. When chyme enters the small intestine, the mucosa is stimulated by tactile stimuli or irritation. It causes the development of local nervous reflexes, which stimulate the glands of intestine. Hormonal regulation. When chyme enters the small intestine, intestinal mucosa secretes enterocrinin, secretin, and cholecystokinin, which promote the secretion of Succus entericus by stimulating the intestinal glands. Methods of collection of Succus entericus. In human beings, the intestinal juice is collected by using multi-lumen tube. The multi-lumen tube is inserted through nose or mouth, until the tip of this tube reaches the intestine. A line is marked on the tube. Entrance of tip of the tube into small intestine is indicated when this line comes near the mouth. This tube has 3 lumens. To the outer 2 lumens, small balloons are attached. When these balloons are inflated, the intestine is enlarged. Now, the intestinal juice is collected through the middle lumen, by means of aspiration. In animals, theory loop. A portion of intestine is separated from the gut by incising at both ends. The cut ends of the main gut are connected and the continuity is re-established. One end of isolated segment is closed and the other end is brought out through abdominal wall. It is called theory loop or theory fistula. Theory Vela loop. Theory Vela loop is the modified theory loop. In this, a long segment of intestine is cut and separated from the main gut. Both the ends of the segment are brought out through the abdominal wall. The cut ends of the main gut are joined. 1. Malabsorption. Malabsorption is the failure to absorb nutrients such as proteins, carbohydrates, fats, and vitamins. Malabsorption affects growth and development of the body. It also causes specific diseases, see below. 2. Malabsorption syndrome. Malabsorption syndrome is the condition characterized by the failure of digestion and absorption in small intestine. Malabsorption syndrome is generally caused by Crohn's disease, tropical sprue, steatorrhea, and celiac disease. 3. Crohn's disease or enteritis. Enteritis is an inflammatory bowel disease, IBD, characterized by inflammation of small intestine. Usually, it affects the lower part of small intestine, the ileum. The inflammation causes malabsorption and diarrhea. Causes. Crohn's disease develops because of abnormalities of the immune system. The immune system reacts to a virus or a bacterium, resulting in inflammation of the intestine. Features. Malabsorption of vitamin. Weight loss. Abdominal pain. Diarrhea. Rectal bleeding, anemia, and fever. Delayed or stunted growth in children. 4. Tropical sprue. Tropical sprue is a malabsorption syndrome, affecting the residents of or the visitors to tropical areas where the disease is epidemic. The cause of this disease is not known and it may be related to infectious organisms. Features. Indigestion. Diarrhea. Anorexia and weight loss. Abdominal and muscle cramps. 5. Steatorrhea. Steatorrhea is the condition caused by deficiency of pancreatic lipase, resulting in malabsorption of fat. Refer chapter 39 for details. 6. Celiac disease. Celiac disease is an autoimmune disorder characterized by the damage of mucosa and atrophy of villi in small intestine, resulting in impaired digestion and absorption. It is also known as gluten-sensitive enteropathy, celiac sprue and non-tropical sprue. Cause. Celiac disease is caused by gluten. It is a protein present in wheat, oats, rye, barley, and other grains. Gluten is like a poison to individuals with celiac disease, because it damages the intestine severely. Features. Diarrhea. Steatorrhea. Abdominal pain. Weight loss. Irritability. Depression.